So the approach that I thought we will take for this one, in fact, and this is where we're going to take advantage of and take the benefit of having Mark join us. Mark actually did a preliminary review of those two substances, basically a review of the GFoRSS assessment associated with these substances, but also a review of what other jurisdictions may have done or may have reached with regards to these substances, in order to help us actually develop our own sort of analysis of these substances and where they stand in the Codex process. So we're going to be looking at agenda items 5 and agenda item 6.1 of the CCRVDF. Agenda item 5 is really looking at the maximum residue limit for Flumethrin. So there was, in fact, a proposal where the MRL was adopted at step 5, and you know what it means. When something gets adopted at step 5, it means that most of the issues were resolved. There were a few things that needed, some pending elements needed to be discussed. With your permission, I'm going to be asking Mark to actually go through the document and summarize essentially the evaluation for Flumethrin, looking really at the various evaluations that were conducted, but particularly focusing on the GFoRSS assessment of 2017 and the pending issues and why CCRVDF actually did not move with the approval at step 5.8.
All right, thank you, Samuel. As has been illustrated in the documents that Samuel provided, we've attempted to give a bit of background information on the chemical under consideration. In this case, they're all veterinary drugs. In this first case, Flumethrin, it is part of the pyrethroid insecticide group. And for the toxicologists in the group, there are a large number of pyrethroids that have been approved as insecticides. In this particular case, it's primarily used for external treatment against parasitic insects and ticks. And it already has food-based approval in a number of animal species. So in this particular case, I think it's relatively straightforward. There's a good agreement international on the health-based guidance value, which in this case was established by GFoRSS as 4 microgram per kilogram body weight per day. And we've provided a little bit of information on the derivation of the health-based guidance value. It's relatively standard. There was a complete toxicology data package submitted in support. So when GFoRSS looked at this, it was primarily to reconfirm the previously established ADI, and then look at some of the intake for generation of MRLs. In this particular case, it's a MRL specific to honey. So in terms of application of this particular pesticide, what we'd also like to point out is all these synthetic pyrethroids are actually established from natural pyrethrins, which are chrysanthemum extracts. So not only are they relatively toxic to a number of ectoparasites, use patterns have to be monitored quite closely because pyrethrins are also very toxic to aquatic organisms, especially fish. So in this particular case, as there really was no issue with the ADI that the JECFA has established, they also looked at developing for a number of veterinary drugs, as they do with pesticides, an acute reference dose, and these are specifically the description of that is being provided in the ARFD description, and based on background information, ARFDs are typically established for any chemical, whether they be a contaminant, a veterinary drug, or a pesticide, where there is sufficient evidence for short-term acute toxicity. So because an acute reference dose and an ADI, which is a chronic health-based guidance value, were established, JECFA then proceeded to give estimates of both chronic dietary exposure for the two main categories of the population, children and adults, and also acute dietary exposure estimations for both the general population and adults or children. And those intake values would subsequently be compared to the applicable health-based guidance values. So in this case, the acute intake values would be compared to the acute oral reference dose, and the chronic intake values would be compared to the ADI. So in this particular case, the MRL that's being recommended indicates that it contributes minimally to the health-based guidance values. In this particular case, it's only 2% of an acute exposure, and let's see, I think it was 6% in developing these chronic intake estimations. As some of you may be aware, it's a relatively standard process where they look at all the available global data that's been submitted to the GEMS, which is the Global Environmental Monitoring System, which has a food-based exposure. And this is something that both WHO and FAO contribute to. So they collect through food survey information about consumption by various age groups in the populations. And this is based on a series of GEMS cluster diets, which the world is divided into. And various aspects of developing these chronic and acute dietary intakes are available from both WHO and FAO. So I guess the gist of this, it's an insecticide which has relatively low toxicity to mammalian species. It's primarily used externally. And this is the same thing with the application in bees. It's not directly applied to bees. It's it's allowed to be infiltrated in the beehive through some strip process, which has been soaked in the pyrethroid. There has been estimations of migration into both the honeycomb and the honey, but in general, these end up being relatively low values. So the proposed MRL was primarily set based on twice the limit of quantification. And typically in the JECFA assessments, they consider analytical methodologies that have been submitted by the sponsors for this particular vet drug. And in this case, a fully qualified method, which is the LCMS-MS, was used in the studies to, and then resulted in the limit of quantification being approximately 3 microgram per kilogram or parts per billion. So typically in these cases, they set the MRL at double the uppermost limit of quantification. So as Samuel has described here, this appears to be relatively straightforward. There's a number of MRLs that have already been established for Flumethrin in various other animal species. There is we would expect that essentially the MRL, even though it was held at step 7 at this particular meeting, will progress for adoption or recommended for adoption by CAC at step 8.
Thanks, Mark. I guess actually the main concern was, and the reason why I guess he did not proceed directly to step 5.8 was mostly related to some comments made by some delegations related to the fact that the analytical method that was used in this regard was an LCMSMS method, considered to be not so accessible to everybody and therefore leading to some potential concern. Now, I guess two elements maybe to mention in this regard. Well, the level that is proposed here, yes, it is based on an analytical approach. It's showing essentially that all the GFoRSS studies showed that there weren't really quantifiable residues that were found in honey after treatment with Flumethrin product. So it seems that there is really a good evidence of achievability of the value that is proposed in this case. So essentially, there isn't an expectation of trade impediments. The issue of potential trade impediments related to the control of the substance, I guess actually, it can be addressed in two ways. Well, first, countries that are producing honey and are exporting it, I mean, they have to show to a certain extent, the satisfaction to the level of safety and quality. And nowadays, I guess private laboratories, for the most part, do have LCMSMSs. And for countries that are importing honey, I guess, they are in a position to require a certification from an analytical standpoint that allows a detection limit that could reach the 3 ppb that is mandated here. So I guess that objection is not a health-related objection, and I don't know that it reaches the threshold of a trade-related objection either, because again, of the propagation of LC-MSMS-based methods. Okay.
Yes, I wanted to just comment here. I think in MENA region, very rare countries that they are testing honey for pesticide in general. This is a major challenge here in the area when we are trying to challenge the limit. Because, whatever comment need to be justified, need to be really, with the data. I'm not sure if there is any data related to GEMS and GEMS related to contamination on honey per country or per region. But what I know in our region, honey has not really been tested very well for pesticide. Very, very limited data about pesticide in general in honey.
Yeah, I think
this
actually,
is a challenge here.
Yeah,
yes.
yeah, I think there is a recommendation there that could be made, at least for the region as a whole, which is essentially that honey is a commodity that is, to a certain extent, quite widely consumed by the population in the Middle East and North Africa. And there might be an interest really to recommend that a residue program, not only pesticides, so essentially pesticides, veterinary drugs, like with target substances, need to be put in place such that we have a better understanding of the level of occurrence of a number of target substances in honey. So that could be a general recommendation that we could make here. I mean, one of the reasons why we're studying these agenda items as well is not only to help, the preparation of the delegations to be there, but also to get recommendations for risk management in general for the region. So,
and just to add to that, I mean, it is a good point because typically, without direct application, you potentially get transfer of pesticides, not necessarily vet drugs, but pesticides based on the production process of honey. So obviously the honey, the bees will pick up the pollen from plants that have been treated with the pesticides, and there's a potential transfer of that to honey. But just to point out is that honey as a commodity is captured by the dietary surveys that are run by WHO and FAO. And although Samuel and I didn't include the values, they do have relatively high acute dietary intakes by various age groups. So for the general population, it's up to 194 grams a day. And for children, it's listed as 90 grams per day. So those would be based on survey results from the various cluster diets that WHO and FAO have developed. If they've picked it up as part of their cluster diet, there may also be residue analysis, including the region where the honey came from and where the residue analysis were developed.
Thank you, Sam. Thank you for these documents. It's very clear, especially for honey. But I think for this veterinary drug, Flumethrin is also observed in bovine products. So my question here, it's also limited only for honey, or we can add bovine products like milk, like muscle, like meat of bovine.
In general, GFoRSS addresses requests coming from the Codex Committee on Residues of Vet Drugs and Foods for specific commodities. And you are correct, Flumethrin has previous approved uses in a number of food-bearing animals. I think it's primarily cattle, sheep, goats, and horses.
Yes, they are here, actually. It's cattle, sheep, goats, and horses, at least for things that can be consumed, you know. So
it is a relatively effective treatment, primarily externally. So it's not applied internally. But MRLs would already have been established for all these, and in all cases, the combination of the exposure based on the MRLs would always need to be below the health-based guidance value.
Yeah.
So in this particular case, the fact that the the honey MRL is actually based on a limited quantification, and there has been no detectable residues. I don't believe GFoRSS actually considered it necessary to go back, and they did develop estimated intakes, but those were based strictly on the analytical limit of quantifications and not residues.
Yeah.
So, but in this particular case, the discussion would only be specifically for Flumethrin in honey.
Yes. I would like to ask about the cumulative risk assessment of pyrethroid. As you know, pyrethroid used in agriculture as a pesticide of agriculture and also as drugs. So there is many sources of pyrethroid as general, not this compound exactly. So how we can consider the cumulative risk assessment of uses of pyrethroids in many compound in food as general?
No, that's a very good question, because as you've indicated, pyrethroids are a relatively broad category of insecticides which have numerous uses. I think the latest estimation is that there has been up to 42 different pyrethroids that have been developed, and some are used primarily for food-bearing animals, etc. Some are used in other veterinary medicine practices. I mean, I think the good point about pyrethroids, if there is a good point for insecticides and veterinary drugs, is the fact that they have relatively low toxicity for mammalian species, and they are much more active against, from a concentration or exposure perspective, against insects based on their mechanism of action. That doesn't necessarily mean that humans, if they're exposed to sufficiently high quantities of pyrethroids, will not show effects. But in general, the toxicity for pyrethroids tend to be minimal for mammalian species. And once again, as we indicated in the document, all pyrethroids are derived from pyrethrins, which are natural compounds extracted from chrysanthemums.
Yes.
So in general, I mean, and the pyrethroid, synthetic pyrethroid categories, as you may be aware, are subdivided based on their mechanism of action into two distinct categories, and they operationally just define them as, you know, class 1 pyrethroids and class 2 pyrethroids, and each have slightly different effects, but the majority of pyrethroid insecticides are subdivided into those two categories. But it is a good point to raise, because in general, any GFoRSS assessment for either a pesticide or a vet drug typically is specific to that particular compound. And the exposure assessment is particular for that ADI and is not considered from an additive basis, simply because, well, part of the reason is that they think it's because all pyrethroids have similar mechanisms of action. They think it would be unlikely that there would be exposure to multiple pyrethroids at the same time. So it's almost, if you're going to use one pyrethroid, then you won't use
the other.
5 or 10 other pyrethroids.
Yeah.
So, but in general, it is a good question to keep in the back of your mind that, first of all, for any individual insecticide, pesticide, or vet drug that has multiple uses, that there would be a cumulative exposure from MRLs from all uses that should be taken into consideration. And in this particular case, because you have a number of structurally related pyrethroids which have approval, there is likely a potential for exposure to multiple residues. But in fact, I think GFoRSS takes the position that this is unlikely to occur because there wouldn't be multiple uses of different pyrethroids simultaneously or potential exposures.
Based on the situation we have here, we have a high level of safety, is during, if you are speaking about 6 microgram per kilo, that's really very low limit based on the situation that we have.
Yes.
But the question is there a possibility other member state from different region will try to increase this limit? Because for us in the region, 6 microgram is perfectly, and I don't think that any from area will object this limit due that we don't have a big industry for export for honey. So we don't expect objection to increase this level. So just the question, is it possible that other member state will increase this limit, and what will be our position if they try to push it to 12 microgram, for instance?
Yeah, it doesn't seem to be the case, actually, Fadi, based on at least previous reports of CCRVDF. I mean, the fact that this was adopted at step 5, per se, the only pending action based on the documentation from CCRVDF seemed to be related to the analytical method. So the only thing that people objected to was potentially the difficulty of accessing the instrumentation that is needed to conduct the analysis.
Yes, Sami. I want just to highlight the fact that honey is a product that we consume a lot in our region. And I think to establish this MRL is very important for us. And I don't know, it's a question that I'm asked for me and for you. If maybe our consumption will affect this limit. The exposure will be the same if we consume a lot this product regarding the issue? Just to add something, Sami, you know that in a certain period, like Ramadan, we import a lot of quantity of this product, and the consumption is very, very high in our country.
Yes.
So, it's a good point because, as we mentioned, as an acute reference dose has been established, the WHO food surveys that they conduct with Codex would consider any national estimates of dietary intake, especially in their, looking at acute exposure. So during specific periods of time that aren't averaged out over a year. And in doing that, they have they had access to survey information from a number of countries, and one of these was Belgium. So a survey of adults in Belgium had acute intake of honey up to 190 grams per day, which is a significant amount. And in
that's a lot of honey to be consumed in a day, to be honest.
So, and then in Spain, they had another dietary survey from children, which specifically measured honey intake, and that was 90 grams per day. So these are relatively high intake values, and those were used in factoring in the fact that an MRL of 6 microgram per kilogram or 6 ppb, even at that level of high intake, provides a minimal contribution to the health-based guidance values. So I would think, even substantial honey intake beyond the 190 grams per day for adults and 90 grams per day for children would still be in the same category, that it would only make a relatively negligible contribution when compared to the available health-based guidance values.
So I'm actually adding some of the points here identified, just to add that to the discussion. So honey being an important commodity for the region, and actually maybe highlighting that the assessment accounts for that, that there is no issue, or that doesn't seem to be an issue for this. So I'm going to be making some changes to this just based on the discussion that we're having right now. So I don't know if there are any other questions for this one. I guess on this one, maybe with some of these changes and some of the additional recommendations, we're going to reach really sort of an analysis for this substance.